also to Sophie and team who give me this great chance to visit Cambridge and also share my work here. So this is the title of my presentation today as introduced. My main target is GIRK channel. GIRK is G-protein-gated inwardly rectifying potassium channel. And this channel could be activated by the binding of G-beta and gamma subunit, which dissociate from the G-protein after its stimulation. And so far, 4 subfamily members have been found, which are GIRK 1, 2, 3, 4. They can form functional heterotetramer or homotetramer, which contributes to the regulation of neural excitability or the rate of heartbeat. As for BD, BD is one of the antagonists of sigma-1 receptor. In my other project, I'm trying to examine the regulation of sigma on M2 receptor. So sigma is one of the chaperone protein, and M2 is one of the GIO couple receptor. So here we use the GIRK channel as the effector of M2 receptor. So the function of M2 could be measured by the GIRK current. So in this project, in the expression of sigma-1 receptor, we apply BD to block the sigma. However, we found that even in the absence of sigma-1 receptor, the BD still inhibit the GIRK currents here. So we are wondering whether this BD could direct inhibition on the GIRK channel or not. So, thus, the aim of our research is to identify whether BD could direct inhibit GIRK channels or not, and to review the structural determinants of the inhibition of GIRK channel by BD. And these are the methods I'm using in this study. I mainly use xenopus oocyte as an in vitro expression system and perform 2 electrovoltage clamp on it. And we also try to record the currents from atrial myocyte by patch clamp. In the latter part, I'm going to introduce some results about the molecular docking using these 2 software which are available online. So these are the results I'm going to show you in the following slides. So let's start from the inhibition of GIRK channel by BD1047. Here we express different types of GIRK channel in xenopus oocyte and measure the basal current of the GIRK channels. GIRK4 always co-express with its natural activator G-beta and gamma subunit for clear observation of its current. And these graphs are the time-lapse change of the current amplitude due to the change of the bath solution. For example, this is in the beginning, this is high sodium, almost no current. When changed to high K, they're showing clear inward GIRK current. And the application of the BD could dramatically decrease the current. And this decrease of the current on GIRK channel could be seen in all 4 cases, suggesting that this BD has direct inhibition on GIRK channel, which is confirmed by statistic analysis. And when you get closer look at the data, you will notice that this BD has most strong inhibition on GIRK4 channel and lowest inhibition on the GIRK2 channel. And to exclude the possibility that G-beta and gamma subunit can also affect the BD's inhibition on GIRK, we co-express with them with these different types of GIRK channel, and similar results was got as left side. So this suggesting the inhibition of BD on GIRK channel is independent of G-beta and gamma subunit. We also had tried another antagonist of sigma-1 receptor, which is called BD1063. And we found it also inhibit the GIRK currents with the same trend on GIRK4 and GIRK2 channel. But today I didn't show you the detail of the results. So as I mentioned, because BD has most strong inhibition on GIRK4 and low inhibition on GIRK2, so we pick up these 2 for further study. The first thing we did is that to make the dose-block relationship of the BD on GIRK4 and GIRK2 channel. Here GIRK2 also co-express with G-beta and gamma subunit for reach the same experimental condition as GIRK4. By applying various concentration of the BD on them, I got the dose-block relationship showing on the right side. The IC50 of this BD on the GIRK4 is around 17 micromolar. As for GIRK2 is around 75 micromolar. So indeed, this GIRK4 is more sensitive than GIRK2 to BD. And we're also trying to check the voltage dependence of this block of GIRK4 channel by BD. What we do is that to measure the GIRK4 current in 3 different bath solutions, and high K, BD 10 micromolar contains in high K or 100 micromolar, and apply this step process and got this IV curve in 3 different conditions. Then we compare the decrease ratio at each given voltage and found the decrease ratio in 3 different bath solutions at same given voltage always similar. So this suggesting that the block of the GOG4 channel by BD is not voltage dependent. And then we try to examine BD's effects in the atrial myocyte. We did this experiment because the GOG1 and 4 endogenously express in atria. So we isolate the atrial myocyte from red and did patch clamp on it. Here, please pay attention to the time-lapse change of the current at minus 100 millivolt, as in shown like here. So in the beginning, we tried the acetylcholine, which is the endogenous ligand to induce the GOG current in atrial myocyte. And you can see a clear GOG-induced current. And then we try to use the TPNQ. This is the specific blocker of the GOG channel. And to confirm this induced current is carried by the GOG channel. And you can see the application of TPNQ blocked the acetylcholine-induced current, suggesting this current is come from GOG channel. Then we start the real experiment and try the BD, start from 0.1 micromolar to 100 micromolar. And we found even the 10 micromolar of BD has strong enough to inhibit the GOG current in the atrial myocyte. The IC50 of this BD in the atrial myocyte is 7.5 micromolar. Then, as the next stage of the experiment, we're trying to identify the binding site of this BD on the GOG channel. By taking advantage of the BD's different effects on the GOG2 and GOG4, we did the chimer study between them. So here, GOG2 has been divided into 5 parts, and each of these parts have been replaced to the corresponding part from the GOG4. And the red letter 4 here indicates this position have been replaced to the GOG4 region, and thus 6 chimers have been made. By electrophysiological recording, we found if the chimer contains the whole N term, pore forming region, or C term, it shows relatively strong inhibition. Especially when the chimer contains the N term, it shows strongest inhibition. So we start to search in the binding site from the N term. Here are another 2 N-term mutant chimers. The difference between them is that one of them contains the whole N-term from GOG4. The other one only contains the distal region of the GOG4. So in the experiment, we found if the chimer contains the whole N-term from GOG4, it shows strong inhibition. When it only contains the distal region of the GOG4, it shows weak inhibition. So combined with this data, it's suggesting that it is not the distal region of GOG4, but this proximal region of the GOG4 on the N term is quite important and responsible for the effects by BD. Then we focus on the proximal N term to search in the binding site. So this is the sequence alignment between GOG2 and GOG4. And this arginine is the starting point of the transmembrane 1. So before that, it is N term. We highlight this proximal N term we found, which is important. And you can find only 3 different amino acid residues. So we mutated all of them and found this isoleucine on the GOG2 is quite important. This is because this isoleucine, after it mutates to the leucine, which is corresponding amino acid on GOG4, it shows acquiring of the inhibition just as GOG4 wild type. And on the other hand, we try the reverse mutant on the GOG4. Here, this leucine 77 has mutated to the isoleucine. And after mutation, it loses the strong inhibition as the wild type. So it confirmed that the leucine 77 on GOG4 is quite important for the inhibition effects by BD. And then I'm going to show you some results about the molecular docking of GOG4 channel and BD. This is the computational docking results. So here is the homology model of GOG4 based on the available structure of GOG2 channel. And this cyan cluster's position indicates possible binding site of BD to the GOG4 channel. And actually at each position, you can see this is not only one BD binds to there, but multiple BD binds to the same position. So it is because the computational docking results will predict all the orientation and direction that BD can bind to there. So it shows in the cluster form. And the higher intensity of the cluster gathering there, the higher possibility that BD can bind to that position. So we highlight the most intensive part. You can see here on the N term and mark this Lewis 77 we found. They have high chance to interact with each other. By playing with the same software, we found some other amino acid residues which near the Lewis 77 might also have chance to interact with BD. And these amino acids you cannot be found by only compare the sequence alignment because they are conserved between the GIRK2 and GIRK4 channel. Then we pick up some like the Lewis 74, 81, or 84 to see what will happen. We mutated to the alanine and found after mutation, all of them loses the strong inhibition. So this indicates not only the Lewis 77, but this region might also involved and important for the inhibition. And this also confirmed the validity of these docking results. Then I'm going to show you some other BD binding site on the GIRK4 channel. So we did this experiment because in our electrophysiological recording and also the docking results, both of them showing that not only N term, but maybe pore forming region and C term, like here and here, might also involve for the inhibition. Thus, based on the docking results, we are trying to search some other candidates on the pore forming region and C-term region. Finally, we pick up these two amino acids on the pore forming region and these two amino acid residue on the C-term region, because you can see they are close with the BD and judging from the direction, so they have high chance to interact with BD. And then we did mutagenesis study on these 4 positions. So here is the detail of the results. After mutation, 2 of them loses the function and the other 2 maintains the function. The important thing is that in the pore forming region, this mutant, E147, after mutation, it loses the strong inhibition, and the other one still has no significant difference with wild type, suggesting that this glutamine acid 147 is critical site on the pore forming region for the GIRK4 for the BD's inhibition. And finally, I'm going to show you some results about the competition between BD and ivermectin on GIRK4. So you may be wondering, what is ivermectin? Ivermectin is the anti-parasite drug which often used in Africa and saved many lives there. And our previous lab member, Dr. Jing, she found that this ivermectin could directly activate the GIRK channel. And important thing is that this critical binding site of the ivermectin on GIRK channel is on the GIRK2, this isoleucine 82. And the corresponding amino acid is the Lewis 77 on GIRK4, which exactly is what we found for the binding of this BD. And although this ivermectin prefers to bind to GIRK2, but it still binds to GIRK4 at the same position. So there might be some competition between them at the same position. Thus, to examine this, what we do is to make the dose-block relationship in the presence of ivermectin and in the absence of ivermectin. And we observe that in the presence of ivermectin, this dose-block relationship shifted to the right side, which suggesting that the presence of this ivermectin lowers the sensitivity of GIRK4 to BD. Also, we try to measure the inhibition kinetics of this BD on the GIRK4 channel in the presence of ivermectin and the absence of ivermectin. What we compare is this inhibition speed at this range, here and here. You can see in the absence of ivermectin, it seems the inhibition is faster to reach the steady state. So we did some further analysis to compare the inhibition kinetics in the same time range. You can see this curve shifted to the right side, and we calculate the tau from this figure, and indeed, in the presence of this ivermectin, the inhibition time becomes longer. So this, again, suggesting that the presence of ivermectin decelerates inhibition speed of BD on GIRK4 channel, reflects some competition between BD and ivermectin at the same position. Here, I would like to summarize my presentation today. what we found is one of the antagonists of sigma-1 receptor, which is called BD1047, can direct inhibit the GIRK channel instead of sigma-1 receptor. And it has more strong inhibition effects on the GIRK4 channel. So by the chimeral mutagenesis study, we identified this leucine 77 is quite important for its inhibition, which is confirmed by the docking results. And in the end, I'm also showing you some competition between the inhibitor BD and activator ivermectin at the same position. This suggests that this position might be a hotspot for the binding of the GIRK drugs or ligands, and might be a good position for designing a drug for the GIRK-associated diseases. Here, I would like to say thank you to all the lab members, especially Dr. Jing, Dr. Tateyama, and Professor Kubo, for their discussion and supporting in my study. So that's all about my presentation. I'm happy to take questions.
Thank you very much for the story and for being in time. So we have time for questions.
Hi, CC. Nice talk.
Thank you.
I was curious about the residues identified in the pore region and the C terminal. Is it possible that they aren't a binding site for the molecule, but rather they're part of an allosteric pathway that when the molecule binds to the 7-7 residue, those residues in the C terminal pore are part of the transduction cascade that brings about inhibition, rather than being binding sites?
That might be possible, I think. Right. Yeah.
Yeah, the reason I say that is because you don't get voltage-dependent block. And if it was blocking in the pore, wouldn't you get voltage-dependent block?
I cannot answer that now, but might be.
Yeah. Anyway.
Yeah, you're right.
I have a question myself. In the previous slide, the leucine, there, 77, is pointing towards the lipids, I would say, towards the membrane.
Yeah.
So, did I understand correct that you found that the binding site is contributed, beside from the leucine, also from other positions in the in the pore domain? Or I mean, I was wondering, how is it possible that also other positions which are on the pore domain contributes to the binding site of BD, even though leucine is pointing outside? I'm not sure I was clear in my question. So,
you mean here?
Is pointing away from the rest of the channel.
You mean the leucine 77 on the end term? Okay.
Yeah. So how can it form a binding site together with the other position that you have identified?
Yes. So maybe this BD is permeable for the plasma membrane. So it's possible if although this faces to the hydrophobic side.
But is this MD based on a crystal structure, on a structure of the channel, or is a model?
It is a model, yes, because this structure is not available.
So you don't know exactly the position of the side chain?
Yes.
Okay. Other questions? Okay, so thank you very much again.